So the question is, could Anopheles gambiae breeding in water bodies with chemical residues and xenobiotics confer resistance as a result of long-term exposure? And that leads me to my aim, which was to understand the effect of adaptation to breeding in polluted water bodies on the development of insecticide resistance. So in an earlier study, physicochemical parameters were analyzed in water from these sites, where the red boxes are showing values that are above the WHO reference guidelines. And when we look at Kolebu, almost all the parameters shows higher values above the WHO guideline, which indicates pollution. Hence, I chose Kolebu as my study site. So for this work, So I collected water samples from Kolebu, which is an irrigation site in Accra, and then I brought it to the lab, and I bred insecticide susceptible mosquitoes that are lab trained in this water. And some of the mosquitoes were taken through DNA extraction to check KDR mutation, and also through biochemical enzyme activity. And some were also taken through RNA extraction to look at gene expression levels of some selected genes. So for my first objective, I was supposed to breed 10 generations of insecticide susceptible Anopheles gambiae in the field water, and that I was able to successfully breed the 10 generation through this process. So my second objective was to detect KDR mutations, and this mutation is associated with insecticide resistance in these mosquitoes that have been bred in the field water. And this is a picture, a gel picture showing how the bands look like. And so after the analysis, I selected first, fifth, and 10th generation to look at, which represents the entire 10th generation. So when we look at the SS, which is the susceptible alleles, we see a decrease in the SS allele as the generation progresses, and also we also see an increase in RS, which is the heterozygous allele. And so we can say that since the mosquitoes have to adapt to this new breeding site, they are selecting for alleles that could allow them to adapt. So the second objective was to look at certain enzymes that have been also associated with insecticide metabolism. And so I look at the first, fifth, and 10th generations as well, looking at the larvae and adults. And in most of the enzymes, I saw an elevation of activity, but these four showed a significant elevation in activity. Also, I wanted to see if comparing the developmental stages, that is the larvae and adults, in the various generations, if I could see significant elevation of enzyme activity. And then looking at the one of the activity or the assay, that's the alpha esterase activity, there was an increased activity level in the adults as compared to the larvae. And this could be explained that since these enzymes have been associated with insecticide detoxification, the adults need to develop these enzymes or express this enzyme in preparation of being able to metabolize insecticide when they are exposed to. But then when we look at the one of the enzyme activity, that's glutathione esterase, we rather saw the opposite, which was showing rather an increase in enzyme activity level in the larvae. And this enzyme has been associated also in the development of the mosquito. And also the larvae also need to metabolize the xenobiotic they are exposed in the water. And then my last objective was to look at some selected genes that have been associated with insecticide metabolism. And I look at these 5 genes. And when you look at the fold expression in the larvae mosquitoes relative to the control samples, we saw that the gene expression decreased with increasing generation. So the various colors represent the various generation, and we are not seeing any green bar here. So it indicates a decrease in the gene expression. And this could be explained that since the mosquitoes' first time exposure to this water body allows them to express this gene in order to be able to adapt to these water bodies. And also when we look at the adults, there was rather the opposite, which shows an increase in gene expression with increasing generation, which reflects an adaptation towards insecticide resistance. And by comparing the expression fold between the adults and the larvae, we saw that the adults showed lower expression, in that since the larvae are the developmental stages that are directly in contact with the water, they might be encountering certain xenobiotics and chemicals in the water, and also microbiota in the water. That's why we are seeing a very high expression in them. So in conclusion, we saw an increase in knockdown mutation as the generation increased, and also the enzyme that have been associated with insecticide metabolism also increased as the generation progressed. So we can say that we can associate breeding in polluted water with development of insecticide resistance. And then with my recommendation, I recommend that the water bodies will be analyzed for different pollutants and insecticide residue, also undertake insecticide susceptibility tests on these mosquitoes to show whether they are resistant to the insecticide or not. So I'd like to thank WAGBE for paying my tuition, and then SEWAT for supporting my research, and also thank my supervisors and these few people. Thank you,
Sandra, for your talk. Thank you very much. And also thank you for keeping it brief, because we're running out of time. Any questions?
Sandra, how did you ensure that your water samples you collected do not contain, they did not contain the wild type, the eggs of the wild type?
Okay, so
because I know the Kisimo strains are highly susceptible to polluted water.
Okay, so to be able to make sure that we did not bring any failed mosquito into the lab, when I brought the water, I kept them for some few days. So if there are any eggs there, they will emerge into larvae, and also sieve the water to make sure I remove any failed contaminants before I use the water.
Okay, thank you very much.
Thank you very much. You have a new pilot, so take care. Seatbelts should be on. Our next speaker should be Isaac Ochere. And for the last two speakers, please, let's keep the tempo so that we finish this day as we started it. So if I stand, please take note that that's a warning that the seat belt must be put on.
Good afternoon. I'm Isaac Dakonchi, and I'm presenting on the Mycobacterium tuberculosis sensostrictus sublineage Ghana. It's associated with drought resistance in Ghana. Very funny. This presentation is cut from the first objective of my PhD thesis, and I'm currently in my 3rd year. So a little background: tuberculosis has claimed more human lives, over 1 billion in counting, in the past 200 years, and this surpasses any single infectious disease, making TB one of the most important diseases mankind had ever recorded. The causative agent called Mycobacterium tuberculosis complex, made up of a group of acid-fast bacilli adapted to causing TB in specific mammalian hosts, even though there are occasional cross-species infections. For the purpose of this presentation, I'll concentrate much on the human-adapted MTBC, which is made up of Mycobacterium tuberculosis sensu strictus, the true TB bacilli. This one is found everywhere on the globe, and then Mycobacterium africanum, which is restricted to sub-Saharan Africa, for reasons not yet understood. Now, the MTBSS is subdivided into 5 lineages, lineage 1 to 4, and then lineage 7. Whereas M. africanum is divided into 2 lineages, lineage 5 and 6. These are some of the major oligotypes of the MTBC that are found in Ghana. The most dominant one is the Cameroon sublineage, followed by the Ghana sublineage, and then M. africanum West Africa 1, followed by M. africanum West Africa 2. The least occurring sublineage is the Beijing genotype, which is most commonly found in the eastern part of Asia. Now, treatment of TB in Ghana is by the DOX regimen. And here we have 4 drugs: isoniazid, rifampicin, pyrazinamide, and then ethambutol, targeting specific metabolic pathways of the pathogen. Even though TB is treatable, the emergence of drug resistance, especially to isoniazid and rifampicin, because these are the 2 most potent drugs in the regimen. They are the 2 bactericidal drugs. So when you have resistance to the 2, we term it MDR-TB. And whenever you have resistance to these 2, the probability that that regimen containing the 2 drugs will fail is extremely high. So emergence of drug resistance, especially MDR-TB, is threatening to make TB, which was once a treatable disease, almost untreatable. And the major culprit for this has been pointed out to be non-compliance to treatment regimen, but we cannot overlook the importance of the genetic background of the bacteria. This is supported by evidence from research done in China and some part of Europe, supporting or reporting of genetic association of the Beijing genotype with drug resistance, especially MDR, and also negative association of drug resistance with the lineage 1. Now, effective control of drug-resistant TB can be achieved by rapid diagnosis of drug resistance and then management with appropriate drugs. However, the most available rapid diagnostic kits, 2 I have named here, which are rampantly used globally: MTBDR+, which detects resistance to isoniazid and rifampicin, and then the GeneXpert, which detects rifampicin resistance as a proxy for MDR. These diagnostic kits were made with information gleaned from genome information that had been accrued from limited collection, and by limited collection I mean by diversity of MTBC. And Ghana, being a hotspot for diversity, harboring 6 of the 7 lineages of the human MTBC, Ghana serves as a very nice place to begin looking at the influence of the genetic background of the bacteria on drug resistance. So the aim of the first objective of my PhD was to define the genetic basis of drug resistance in Ghana. And to do this, I had to answer this question: what are the forces driving drug resistance in the country? And I put up this hypothesis that the genetic background, the origin of the strain shouldn't have any effect on drug resistance. So I set out to test for this hypothesis. And my sample sites were northern Ghana, represented by the Mamprisi East District, and then the southern Ghana, represented by the Greater Accra Sub-Metro. And we recruited consecutive TB patients reporting to these selected hospitals between 2012 and then 2014. And in total, we recruited 1,774 patients, out of which we isolated 1,490 TB strains. These strains were tested for resistance or susceptibility to isoniazid and rifampicin using the microplate alamar blue assay, which is a chromogenic assay. Here, I will just put it simply. When you see a blue dot, it means there's no bacteria growing there, so it's susceptible to the drug. And then the pink dot, depending on how you see colors, shows that there's an activity, a bacteria is growing, so you have a change from the blue to the pink. So on this plate, this is an anti-cell plate. Each plate takes 3 strains. The first strain here, you have a isoniazid column, the rifampicin column. There was not a single dot there. And how do we interpret these results? We have the cutoff point. For both drugs, the critical concentration of the drugs by international standards is 0.25 microgram per ml. And that is, so that's the third dose. So here to be 1, 2, 3, somewhere here for isoniazid and rifampicin will be here. So for the first strain, since we don't have any red dot there or pink dot, it means this strain is susceptible to the 2 drugs. The second strain is highly resistant to the 2 drugs because both rifampicin and isoniazid will having the pink dots. This is the negative control and the positive control is down here. Then the third strain, resistant to isoniazid but susceptible to rifampicin. So among the 1,490 strains, those that were resistant to a specific drug, specific genomic regions were amplified and then sequenced for mutation analysis using H7RV as a standard. H7RV is a standard TB strain, pathogenic but susceptible to every known TB drug. So we normally use it for mutation analysis for comparative studies. Now, results and discussions. In total, there were 161, comprising approximately 11% of the 1,490 that were resistant to at least one drug. There were about 7.5% resistant to only isoniazid, about 0.7% resistant to only rifampicin, and then 2.6% MDRs. When stratified by geographical region, that is the northern and the southern part of the country, we didn't find any statistical association, even though the northern part had proportionally higher resistance as compared to the south. Then we stratified the resistant strains by species. I must state that even though all the samples worked on were human samples, we isolated 10 bovine strains, M. bovis. And they interestingly showed higher resistance if you compare the proportions with the human strains. But for the sake of this presentation, we zoom in into just the human adapted MTBC. And here too, we didn't find any association with any species. But when we further went down to the sublineage and lineage level, we found that the Ghana sublineage was highly associated with drug resistance when compared to the Cameroon. The Ghana is the second most dominant type or sublineage in Ghana, followed after the Cameroon, which is the most dominant. And you find association at all levels, be it isoniazid monoresistant, MDR, rifampicin monoresistant, or resistant to at least one drug. When we compare the Ghana sublineage to the M. africanum, West African one, the only association we find is with resistant to at least one drug. This suggests that the two may have comparable propensity to develop drug resistance. But when you compare to M. africanum, West Africa 2, again, you are finding the Ghana more resistant to the drugs as compared to the M. africanum, West Africa 2. Now, this table is a summary of all the mutations that we identified from the mutation analysis. We detected mutation in all targets except the RPOA, which codes for the alpha subunit of the bacterial polymerase. And for isoniazid resistance, the most dominant mutation we found was amino acid substitution at locus 315 of CAT-G from serine to threonine. This mutation is known to encode high-level isoniazid resistance. And then that was followed by a nucleotide substitution at the promoter region of the INHA gene, accounting for about 15.9% of the isoniazid resistance. But something interesting that we found in this research work was that there's this mutation here at the locus 8 of the INHA promoter. This mutation is said to code for drug resistance using the MTBDR plus. But when you come to our work here, we find it not to be associated with drug resistance because all the strains that were having this mutation also had a CAG mutation, which is known to code for high-level isoniazid resistance. For rifampicin resistance, all the mutations that we detected were also detectable using the MTBDR plus. We identified a number of novel mutations. Some of them, especially the one I've highlighted here, has been hypothesized to be a compensatory mutation, compensating for the fitness cost associated with the high-level isoniazid resistance coded by the CAG 315. There were genetic associations with some species, and here what we found was that the MTBSS is associated with CAG mutation, whereas M. africanum is associated with INHA promoter region mutations. And there was no association with rifampicin mutations, associated mutations, but we also found that the proposed compensatory mutation is associated with the Ghana sublineage, which may explain its preference for drug resistance. So in conclusion, this study reports the first association of the Ghana oligotype or genotype or sublineage with drug resistance in the country or in any part of West Africa where it's dominant. And we also reported of possible sublineage-specific epistatic interaction between drug resistance conferring and compensatory mutations, which may have effect on transmission of drug resistant strains in the sub-region. Whereas MTBSS is associated with CAG mutation, M. africanus is associated with INH promoter mutations. This confirms a previous study done by Homoka et al. In 2010. The novel mutations that we identified may have ecological relevance for transmission of drug resistance, and they need to be looked at again in details. And then the most interesting thing is that the hypothesized compensatory mutation was found in only MTBSS strains of lineage 4 harboring the CAG 315 mutation. This may explain the preference of the CAG mutation by this species. We find association between the MTBSS. And on the lighter note, this work supports the use of the MTBDR plus for diagnosis of DLTB in Ghana, but not much when it comes to GeneSpec, because GeneSpec is a proxy for diagnosis of MDR, because we found that most of our isolates were resistant to isoniazid, but they were susceptible to rifampicin. So if you are going to use the GeneSpec as a proxy for detecting drug-resistant TB, it means all these strains will be detected or diagnosed as susceptible. And when you put these people into any regimen containing isoniazid, it may not work. It may negatively impact the regimen. I would like to, last thing before I leave. In order not to be selfish, all the information gleaned from this work had been published. It just came out last month, so you can go and read it and get into details of it. I had to super summarize it here. And I would like to thank my supervisors, Professor Yeboah Menu, Professor Sebastian Gagnon, Dr. Jim Brownford, and then the Professor Yeboah Menu Research Group. Thank you.
Isaac has actually completed all his time, but if there's any one burning question, we can take that. Any burning question? There's none. So at least you predicted that. So we'll move to the next presenter. Thank you very much. So our next presenter is Yao, and I hope you'll complete it on a high note. I come from Kenya, where we have marathon runners, and they usually say, when you are going for your last lap, you assume you do it as the first lap.
Okay. So thank you very much for allowing me to close the meeting, possibly scientifically. That's the last talk. Yeah. So my mentor is Dr. Wandari. So officially I work on invasion. So yeah. So to summarize what people have presented on malaria, over the decades efforts in controlling it, we see a gradual decline in the numbers of deaths that are associated with it. But irrespective of this, there are still roadblocks. which is the development of resistance by the parasite, and also the addition of new members into the species that can infect human beings. So being an invasion person, we set out to look for genes that are involved in merozoite invasion. And for this, we scan plasmodB for genes that harbor signal peptide transmembrane domain and is highly expressed at the late stage of the parasite development. So we did a scan, a number of genes came up, and we took them for characterization. And so I'll be presenting on one, so basically this is a gene which is encoding for 899 amino acids, so approximately it's 106 kilodalton protein. It harbors a single transmembrane domain, and it's a type 2 transmembrane domain protein. So if you do homology mapping across all the species of Plasmodium, you find that it is 80 to 90% conserved in terms of amino acids. But then in the first 200 amino acids, you find the similarity is just 5%. It's very varied there. So we had a database in the lab which had profiles across all the other species in terms of gene expression. So we went to find what this gene looks like as one of the criteria for selecting it. So we see it in the ring stage, and then it comes up also at the late stage at Shizon. So we took it up for further studies. So normally as a gene, a hypothetical gene, we don't know its function. You go and throw it into different informatic platforms, find something that you lay hold on as the basis to now go down to look at the function. So looking at the topology, we realize that the transmembrane domain is there. Then if you blast the whole amino acid sequence, you see this domain come up as a gene that harbored the beta-lactamase domain. But one of the criteria for selecting it is having a signal peptide. But here we don't see a signal peptide, meaning that it's one of the genes that drifted off. So we went on to see, in terms of amino acid similarities with humans and other organisms which already have been characterized to have the beta-lactamase domain. So